Welcome to AP Biology. Our first video in genetics is going to be about the differences between prokaryote and eukaryote DNA. Prokaryotic DNA is highly expressed. About 90% of the DNA gets expressed into proteins. Eukaryotic DNA, not so. We have about 20,000 genes. Some of those can be used to make more than one different protein, but only about 1 to 2% of our DNA actually gets used to make protein. A lot of the rest of the DNA is used for control purposes, and some of the DNA we just don't know what it's used for. In a prokaryote, you can see we have DNA that's kind of looks like a roller coaster. It definitely, if you follow this around, it ends up just being one big loop. And so we call it circular. If we could pull it out and pull it tight into a single line, it would be a big circle. In addition to that, bacteria also have plasmids. These are little loops of DNA that can be used in exchanging genes between different bacteria. This is conjugation is like the one sort of like sex activity that bacteria can do to swap genes. These are loops of DNA from bacteria. These are taken with an electron micrograph, so you can, or electron microscope, so you can see much closer than you normally would be able to with a light microscope. You never see our DNA in the linear form unless it's taken with an electron microscope. You can see chromosomes, human chromosomes, when you are looking at through a light microscope, but we are definitely seeing them in the condensed form. So our DNA, when a cell gets ready to divide, will loop around histones, and then it will coil, and then it will supercoil, and then it does some super duper coiling to form chromosomes. And this is what we actually see during mitosis and meiosis, is a condensed version of the DNA. It's not the linear DNA that we actually use to make our proteins. This is what it looks like with a scanning electron micrograph. So with a scanning electron microscope, you take a picture, and then they have all kinds of techniques now to artificially add color. This would definitely not be blue or gold, but by adding color, you can see the features a little better of the micrograph. We have in our bodies homologous pairs of chromosomes. Bacteria don't. They have a single loop of DNA that gets copied when they reproduce. We have homologous pairs because we are getting DNA from two different parents, whereas bacteria does not. They're not sexually reproducers. Sometimes they swap some DNA through conjugation, but generally speaking, they're just reproducing by binary fission and forming two from one. In us, we have homologous chromosomes. You get one of these from your mom and one of these from your dad, and they are different from one another. They're very similar, but there are some differences between the two. And we talked about that during meiosis, during crossing over. Some of this can be swapped when we make gametes, and these things are going to line up independently during independent assortment. This is how our DNA normally looks. We have single copies of the DNA, so this would be a homologous pair. But you never see it like this because we only see our DNA during replication or during cell division. And prior to cell division, the DNA replicates. And so you always see DNA or chromosomes like this with two sister chromatids attached to each other. But in the G1, which is what the cell is normally in, this is how the chromosomes would be in the G1 phase. It's only after S, when you roll into G2, that they look like this. And then when they condense, that's when they actually become visible like this during mitosis. This is a karyotype of human chromosomes. We have 23 pair of chromosomes, so we have 22 autosomes and then a pair of sex chromosomes. And these store all of our genes. And when you make gametes, you're going to give one chromosome from each pair to each gamete. So they will be mixed up because of crossing over and independent assortment, but you only give one of each pair to each gamete so that when the gametes come back together, you end up with the correct number of chromosomes.